Hi everyone, this is Tuval from Element Biosciences, and we're going to spend the next 5 to 10 minutes learning about how we're applying LoopSeq synthetic long-read sequencing for the sequencing of immune repertoires and how we're deploying it in the antibody development process. First of all, a word or two about the antibody development process. It involves essentially an iterative cycle of designing and building immune repertoire libraries based on either naturally occurring immune repertoires from animals or from humans, or on computational prediction of what productive antibodies are going to be, and those are typically made using completely synthetic DNA libraries encoding antibodies. Once those are built and tested, really the method of testing them, or I should say the method of testing the result of binding them to their target, is typically DNA sequencing. And what we're going to focus on is how LoopSeq synthetic long-read sequencing is enabling this process of antibody sequencing throughout the process of antibody development. So there are essentially three types of sequencing applications that support the antibody development process that involve immune repertoire sequencing. One is simply bulk B-cell sequencing. So this is the sequencing of heavy and light chains individually from bulk preparations of B cells. And we'll learn in the next slide how long-read sequencing is enabling this type of sequencing. The other type of sequencing is single B-cell sequencing. This is essentially the same type of sequencing that we're doing here, sequencing heavy and light chains, but the added layer of information that we're getting here is the pairing of the heavy and light chains, the correct pairing of the heavy light chains in nature, which informs the antibody development process. So both of these types of applications are really applications that inform us about the immune repertoires, and they kind of guide us as to which antibodies we want to build. Once we figure out which antibodies we want to build, the method of testing them usually involves some sort of fused antibody format. Maybe the most well-known one is single chain FV. This is where the heavy and light chain are fused into one polypeptide chain at the DNA level, and then tested typically using phage-display technology. And so this is the third sequencing application, and this also involves long-read DNA sequencing. There are multiple advantages for doing this with long-read sequencing, and we'll learn about them in the next few slides. All right, so focusing on the sequencing of heavy and light chains, really the challenge is that short-read sequencing, whether you're doing paired in 150 or paired in 300, really isn't long enough. The read length is not long enough in order to fully cover the diversity encoded in, specifically in heavy chains. So if this is a read from an immune repertoire sequencing library, you can see that there's the insert size, which is typically the heavy chain, and then there's read 1 and read 2, which read from opposite directions, right? It's a paired end read. And then what happens in a certain fraction of the heavy chains is that these two reads are simply not long enough to fully cover the length of the read, and then you have this inner distance here that is not covered by the paired end reads, and you end up with a certain fraction of the heavy chains that are simply not fully covered by your sequencing, and that's potentially an important part of the diversity that we're completely missing out on. Another problem with short-read sequencing and sequencing heavy chains is that even for inserts where they are short enough to be covered with paired end sequencing, the quality of the short-read sequencing tends to decline towards the end of the read. So this is the region where we're trying to merge read 1 and read 2, but this is also the region where the quality scores of read 1 and read 2 are at their lowest. So trying to merge those reads here when you have a very low quality score is also challenging. And I guess back to my first point here, what you see here is a graph from this 2019 publication showing essentially on the y-axis, the fraction of reads, right? Reads that can be merged, right? Read 1, 2, and read 2 that can be merged as a function of where we're placing our primer on the actual transcript. So you can see that, so the further the primers are from the start of the gene, the fewer reads can be merged. And obviously, as you're planning your primer to be closer and closer to your target, you're missing out on additional information that could have been encoded in that transcript if the primer if you had complete design freedom over your primers, and we'll go over examples here. So these are kind of the disadvantages of using short-read sequencing for the sequencing of heavy and light chains. For example, one type of information that you're missing on when you're placing your primers specifically in a place that would maximize the reads that can be merged is, for example, isotype information. So isotype information in antibodies is the type of information that you'll be losing if you're placing your primers, if you have limitations on where you can place your primers due to the limitations of short-read sequencing. That's one type of limitation. The other type of limitation is what we mentioned before, that some fraction of the heavy chains will be simply too long for read 1 and read 2 to merge, regardless of where you place your primers. The next type of sequencing application we'll discuss where long-read sequencing is beneficial is in single-cell sequencing. So in single-cell sequencing, specifically on the 10x platform, but really on any single-cell platform, enables us to add an additional layer of information to antibody sequencing, and that is essentially the pairing of the heavy and light chains. But it suffers from the same limitations that just standard bulk short-read sequencing of heavy chains suffers from, which is the inability to pair most of the reads and missing out on isotype information. So sequencing this with long reads enables you to get both the paired heavy and light chains, fully isotyped reads, full-length reads starting from the very 3' to the 5' of the gene. You can place your primers anywhere, and you get an extremely accurate long read that is a full-length long read with no gaps and a uniform quality score throughout the length of the read. So you're getting 100% coverage of the heavy chains and the heavy and light chain information. I should mention that with the 10x prep, we're also getting the 10x COMI and cell barcode reconstructed as part of the long read. So you'll be able to demux or sort on those long reads and get information about which cell they came from and which 10x COMI they were tagged by. And then this process of pairing heavy and light chains really informs us about which antibodies we want to build and when we're testing antibodies for clinical development. These are typically made into single chain FVs, which are then tested in phages. So this process is called panning, where the antibody is displayed, then bound to a target, and then we typically sequence the variants that had high affinity to the target. And this is, again, one of the largest advantages of long-read sequencing in immune repertoire sequencing, and specifically in antibody development, is that these single-chain FVs are typically anywhere between 800 and 900 base pairs, which is way too long to be sequenced using short-read sequencing. Even if you're doing paired in 300, you wouldn't be able to sequence even a single-chain FV start to end. So this is where long-read sequencing in the form of LoopSeq, synthetic long-read sequencing, kind of comes to the rescue. And what we're doing here is we're sequencing full-length single-chain FVs. So these are single-molecule long reads that are 900 base pairs long and highly accurate. And if you're interested in how we're reconstructing long reads from short reads using LoopSeq synthetic long-read sequencing, I recommend watching one of our other webinars that take a deeper dive into how LoopSeq chemistry and software actually works. In the context of phage display and panning, in traditional experiments that do not employ long-read sequencing, one would have to subselect the variants that were high affinity binders to the target and choose, say, 96 or 1 or 2 plates of 96 isolates in order to test what the sequence of those binders is. In contrast, because we can do long-read sequencing, we can circumvent cloning altogether and sequence directly the library of binders or non-binders. And we're not limited to 96 colonies because we're sequencing single molecules, so we're circumventing cloning and directly sequencing the DNA encoding antibodies that were bound to the target. So we can sequence thousands, hundreds of thousands, and even millions of variants, and we can sequence both the binders and the non-binders. This gives you much more comprehensive data about your binding experiments and informs the next rounds of panning much more systematically. So this is just a high-level overview of how our system looks like. Input into our kits include either immune repertoires in RNA format or DNA format, single-chain FVs, single-cell experiments out of a 10x instrument, or just phages out of a panning round. Those are used as input into our kits. Our kits have all the reagents that you need to make a sequencing-ready library that can be sequenced on either an Element sequencer or competitor sequencers. The data that comes off of the sequencer is uploaded to our software, which then reconstructs long reads from short reads and makes highly accurate long reads written into FASTQ files. This is a publication from 2021 comparing the accuracy of loop long reads compared to other long read technologies, and you can see that at essentially every possible length, LoopSeq has a much higher fraction of error-free reads. How is this relevant to immune repertoire sequencing is shown here, where we're superimposing the length of different types of immune repertoires on top of this graph. So you can see that even for the shortest format, which is heavy chains, the fraction of error-free reads is much higher for LoopSeq long reads than any other long read technology, and that's also true for single-chain FVs and even for CAR-T molecules, which are much longer. With short read sequencing, you can see that you can typically sequence things that are at the length of maybe light chains and some fraction of the heavy chains, but you're definitely missing on a significant part of the diversity in heavy chains, and you're completely unable to sequence fused antibody formats like single-chain FVs, which means that you'll now have to go through the bottleneck of cloning and sequencing a very small number of variants. If you'd like to learn more about how we're applying long-read sequencing to immune repertoire sequencing and to antibody development or to any other domain in genomics, please visit us at either the Loop Genomics Learn site or at Element Biosciences. Thank you for your attention.